All right, moving into part 2 of chapter 12 in the cell cycle, we're going to be working on mitotic spindle and cytokinesis and some other aspects of division in this section. So let's start with that mitotic spindle. It's a microtubule structure that has in mitosis has two centers, but actually we're going to look a little closer at the organelle during G0 and G1 to show you how it can change and progress when we move into mitosis. But it's made of microtubules, and these microtubules have a polarity to them. They have a plus end and a minus end, and that becomes very, very important when it comes to organized movement of materials, whether we're in cell division or not. So we start with an organelle known as a centrosome. It's an organizing center. It's composed of proteins and tubulin, that's the monomer of a microtubule, and the centrosome is actually at right angles to itself. So we're going to see some pictures of that. We do replicate it during when the cell has been signaled to divide. We do replicate it in order to prepare for that division. And when the mitosis phase ensues, the centrosomes go to opposite ends of the cell and begin synthesizing microtubules to prepare that spindle. And so here is a mitotic spindle where you have centrosomes on either side of the cell. It is comprised of connecting microtubules, these interpolar ones that don't seem to have much of a movement function per se as a stabilization function. Then you have what are called kinetochore microtubules, and like the interpolar ones, the kinetochore ones though have a specific purpose of separating the chromatids and moving the subsequent chromosomes to these distal poles. And then these third ones called aster microtubules. And aster is another like asterisk. Have you ever heard that? An asterisk is a little star. And these aster microtubules are not connected to one another, but they seem to help promote the resulting daughter cell shape. They tend to kind of be out there and pushing against the cell membrane in order to begin and ensure a proper cytokinesis. So we have this centrosome, this microtubule organizing center. And like I said, it has these two kind of bundles of proteins and microtubules. These bundles are called centrioles, and they are at right angles to each other. They're perpendicular to each other, and they radiate a three-dimensional array of microtubules. So here's a nice picture. You can see here again, these centrioles are actually sort of microtubule, a superstructure, a macrostructure of microtubules in this sort of stellar pattern, if you will. But again, they're at right angles to each other. And we're not seeing the proteins that are involved in organizing these microtubules this way. Like I said, it replicates during the G1 phase, and it will be prepared to migrate during the early phases of mitosis. This video right here, which I hope will play, if not, I'll make sure that there's a copy of it in your on ACE for you, is showing the dynamics of a microtubule organizing center, the centrosome, which is centered right here in the middle of this picture. What you're seeing there are the microtubule heads of microtubule synthesis. Then we hit it with an inhibitor, and very much to our surprise, even just a few seconds after we hit it with the inhibitor, those microtubules began functioning again. So that was a surprising finding for us, and we actually published on that. But that's the microtubule organizing center with synthetic heads of microtubule synthesizing proteins moving out out into the distal periphery of the cells. And here you can see the centrosomes functioning in various stages of mitosis to form these mitotic spindles. And you can see the aster microtubules right there, really prominent there during late telophase. So it's harder to see the kinetochore and the interpolar ones, but you can certainly see the asters in this video. Just another set of pictures for you to reemphasize that you need to know the three different types of microtubules that are involved. We have the interpolar here in blue, we have the kinetochore in black, and we have the aster microtubules in pink or magenta. The function of microtubule spindles, what happens is the chromosomes, these duplicated chromosomes, these doublets, are hooked up to the kinetochore spindle microtubules. There is a protein complex on the chromosome called a kinetochore, and that's the attachment point to the spindles. And so what happens is during prometaphase, we hook up those kinetochores to the spindle microtubules and we begin to see movement of the chromosomes. The chromosomes are getting organized for separation. So those kinetochores are proteins that will actively work with the microtubules in an enzymatic way. At metaphase, and we see all the chromosomes line up in what we call a metaphase plate. It is an imaginary structure. There's nothing there, but what we see visually is a lining up of those chromosomes, and that's what we call the metaphase plate. So here's prophase. We're starting to see some condensation here. This is when those microtubules would be migrating to opposite ends of the parent cell. Then during prometaphase, we would begin to see those chromosomes being hooked up to the microtubule spindle and being moved around until they are all moved to a unified sort of center location, and that defines metaphases when we see that metaphase plate right there. So there's a nice picture of metaphase, and you can see the chromosomes are very large relative to this metaphase plate structure. So here's our doublet chromosome. Here's our enzymatic kinetochore. Here are the microtubules. And this is what we would call a metaphase chromosome. It hasn't yet been divided. At the end of metaphase, and what we call in the process of anaphase, we split these doublet chromosomes and they begin to migrate away from that metaphase plate. Anaphase, the next phase in cell mitosis, is when we have separation of the sister chromatids. So they're all lined up at the metaphase plate, and when that separation ensues and the migration of those sister chromatids, now called chromosomes, that's what defines anaphase. Now, one thing we know about the motion of the microtubules is that it is not a pulling motion by the spindles. It's not a pulling motion by the asters. It is actually an enzymatic function by the kinetochore, and that's why I said enzymatic earlier. The kinetochore is actively breaking down the microtubules, and so those microtubules are shortening, and that's what's drawing the chromosomes to the distal ends of the parent cell. So here we have a root onion tip, and we can see the chromosomes migrating. An even better picture here with the mitotic spindle. And what's happening in one little piece of data, notice how much more intense the green is up here versus the blue. Now you might think, well, maybe that's covering it up. You can see a few strands in there, and those are the spindle, those are the interpolar spindle fibers, microtubules. But with the chromosomes, however, we have some evidence to show that the kinetochores are enzymatically depolymerizing, the microtubules. So how do we know this? How do we know this? Well, in order to create the spindle, the microtubules have to be synthesized. And this type of an experiment is known as a pulse chase. That is to say, we can add material to the cell that has a label to it at just the right time. So we can sort of get the the cell to label, use labeled material. So what we see is that in a period of time the labeled material is there, and it'll get incorporated into the microtubules, so you can visualize it. And what we if it was a pulling by the aster, what you would see is that label would move closer and closer to the aster. But what we don't see is that label moving. We see the chromosomes moving, but we don't see the label moving. So when we do this pulse chase experiment, we pulse it with a label. That means that the microtubules that are synthesized get that label, and then we take the label away, and so we have just a unique band here. But what we don't see is that band moving closer to the spindle. And that's why we believe that this process here is microtubule shortening rather than pulling, like say pulling on a tether. The other thing that we can see following that pulse chase experiment is that as these things shorten, that labeled tubulin becomes solubilized. Instead of being stuck in the microtubule, we see labeled tubulin subunits beginning to appear in the cytoplasm, and that reinforces and corroborates the idea that this kinetochore is actually moving the chromosome by actively depolymerizing this end of the microtubule and pulling it along the microtubule in that way, rather than the pulling being from the aster. On the other hand, those interpolar microtubules that are there, they're not getting depolymerized by the kinetochores because they're not hooked up to the kinetochores. On the other hand, they seem to be, or in the opposite way, they are actually pushing. They seem to be involved in the pushing of these asters away from each other. So if anything, they're actually getting longer and generating a pressure motion to push these mitotic asters away from each other. And that's what leads us into telophase. When the chromosomes arrive at this distal location, we see subsequent events to not only push the asters away, but also begin to condense this middle region. And this is true mostly in animal cells. In plant cells, we have a slightly different process. But cytokinesis, that physical separation of the two daughter cells, begins late anaphase and or during telophase, when we're pushing and disassembling this mitotic spindle. Microtubules are certainly involved in this. As we said, those interpolar spindle fibers, those interpolar microtubules are pushing away. But there's also actin and motor proteins involved in the creation of the two physical daughter cells. The actin will rearrange itself somewhere analogous to the metaphase plate, somewhere along that equator, and will begin contracting, forcing material to either one side or the other. We call that a cleavage furrow with a contractile ring of actin microfilaments. So that cleavage, that separating of the two daughter cells, is a physical process involving certainly the interpolar microtubules, but also actin and motor proteins. So we form that cleavage furrow. In plant cells, it's different. You don't have that cleavage ring. What you have instead are organelles that are actively synthesizing cell wall. We call that a cell plate. So here's an animal cell with that contractile microfilament. That's actin and motor proteins. Squeezing that cytoplasm, forcing organelles and nuclei to opposite ends. In plant cells, what we see are vesicles that have enzymes that are involved in forming cell wall, and those vesicles will fuse, and you'll polymerize the polymers, the glucose polymers. Okay. and that creates the new cell wall. Here's a zoom up of that electron microscope. Just to review a little bit, try to get you to take it to the next level when it comes to the steps in mitosis. In prophase, we see partially condensed chromatin. When we get to prometaphase, we see fully condensed chromatin, and we can't see it in this photo, but we begin to form a mitotic spindle, and those chromosomes begin migrating, eventually forming a metaphase plate, which is the hallmark of metaphase. The hallmark of anaphase is when we begin to see the kinetochores separating the sister chromatids, depolymerizing the mitotic spindle fibers, and the migration of those chromosomes. Late anaphase and telophase, we see cytokinesis. In this case, this is the root onion tip, so we're going to see that cell plate because that occurs in plant cells. And so we have cellulose being formed from vesicles, polymerizing glucose to create that cell plate and eventually create two daughter cells. There's a really, really great video, I think, on Khan Academy that you guys might want to take advantage of if you need a little more assistance with this. It does take a little bit of time and a little bit of practice to get all of the details down. As much as I would love you to be able to get it and hang on to it like you would hang on to somebody's name or something, it's never quite that easy because there are more details, there's more synchronous events going on. So Khan Academy is a wonderful resource for you if you would like a different take on cell division. We are talking about the same things, but they just have a slightly different presentation using anagrams and phonics to help remember some of this stuff. Now that we've done eukaryotic, I want to talk a little bit about binary fission in bacteria. And the reason is we're going to kind of bring these two bookends together. We have that eukaryotic cell division, and we have in-betweens. There are in-betweens. We're going to show you that in just a little bit. But in the prokaryotic situation, they will duplicate that circular chromosome, and it actually requires a special set of proteins to separate those two, because when you synthesize two circular chromosomes, they actually end up being linked like in a chain. So we have to separate those chromosomes, and then we see this pinching and restructuring, kind of like the formation of a plate, not as extensive in terms of vesicles as what we would see in the plant cells. Nevertheless, we do see this cell wall forming and growing in between what will become the two daughter cells. We call this binary fission because we end up with two daughter cells. There are other types of fission out there that result in asymmetrical division and so forth. But when we say binary, we're talking about two daughter cells. One interesting thing, and this gives us some insight into eukaryotic DNA replication, is that the chromosomes have this, the bacterial chromosome has this special spot, a special region of DNA called the origin of replication. It doesn't just start randomly in places, though in eukaryotic it seems like it's starting in random places, but what we think in eukaryotic replication is that you have these little special short sequences that are easier to separate than other parts of the DNA. And so in the case of a bacterial chromosome, it's a very, very specific origin of replication. We found this out the hard way, and I say scientists found this out the hard way because they tried to use these little donuts of DNA to insert into the cell, but they wouldn't replicate because they didn't have an origin of replication. But once you put that little origin of replication in there, yes, they can divide quite easily and replicate quite easily. So we have this duplication of the chromosome, the separation of the chromosome, and the pinching of the plasma membrane, dividing the cells in two in binary fission. So here is the series of images from your book. And when an E. coli cell makes the decision to undergo binary fission, the first thing that we see is the formation of two replication forks. Here's one on the left and one on the right. As the DNA is being replicated in this direction and in the other direction, ultimately resulting in two copies of the chromosome. So now we have two copies of the chromosome. We have now you can, if you have a probe for the origin of replication, you can see two locations of that now once that replication has proceeded. After replication is completed and we have physically separated the chromosomes, then we begin to see that pinching and formation of cell wall to complete binary fission. Once we have those two daughter cells, now we have two independent organisms. Remember that prokaryotic cells don't have asters. They don't have microtubule organizing centers. They don't have organelles. All of the functional proteins are either located on the interior surface of the plasma membrane or are floating around in the cytosol. And as I mentioned in a previous slide, we can see the binary fission. So we have where the plasma membrane seems to be involved in the organizing of the chromosomes, moving them to opposite sides, to everything in between. We have lots of in-between sorts of division that look like a cross between the ancestral state and the eukaryotic state that we see now. An example of those different organisms. For instance, here's bacteria up here, true prokaryotes. We have the sort of in between prokaryotic and eukaryotic. It does have a nuclear envelope, but it still sort of resembles binary fission. Certain yeasts, which are true prokaryotes, have a sort of, again, a sort of an in between the binary fission and the true mitosis that we see in higher order organisms like humans or mammals or plants. Lastly, please understand that this mitosis is a very, very complex set of multiple signaling pathways and multiple regulatory proteins. These proteins are enzymes, they are cofactors, and they regulate each other. We have a family of proteins called cyclins and cyclin-dependent kinases. Sometimes we abbreviate those CDKs. Depending on the cell type, we see different patterns of expression of the molecular regulators of cell division. So depending on how often the cell divides, we see patterns of these regulatory proteins. We can force cells to divide if we cause expression of these proteins. And one of the things we've found is that cancer cells, for instance, seem to lose that ability to control. They get overexpression of certain forms of these regulatory cells, pushing cells to divide even when they are not following a normal sequence of events. In fact, in advanced cancers, you actually get this sort of asymmetric division going on because the cytokinesis and the mitosis seems to have uncoupled. In normally dividing cells, this control system almost behaves like a clock. Okay, it has this sort of temporal arrangement of expression of different proteins that is controlled and influenced both by internal signaling as well as external signaling. Most cells that are undergoing division require an external signal to say, hey, you need to keep dividing. And let me give you a sort of example of what I'm talking about. Let's say you accidentally cut your finger and you have a wound there after the bleeding is under control. Well, the cells are now in that area have been stimulated to grow and reconnect with each other. And that feeds right into this cell cycle control system. Once the cells have grown back, assuming it's not too severe of a cut, you have a resumption of a contiguous epithelium that you can't really see any sign that there had been a cut there before. That's because what's happened is those cells have regrown until the point that they have achieved that continuous epithelium, and then those checkpoints kick in and prevent the cells from growing any further. That's the point. The checkpoints can initiate cell cycle progression, but can also restrict cell cycle progression. And so we have sort of three major checkpoints. We have this one right here. Anything over here, if the cell doesn't get past that G1 checkpoint, it's in what we call a G0 state. That is to say, unless it knows that it's ready to move through into DNA synthesis, it will stop at that G1 checkpoint, unless there is a signal to grow. And then we have our S phase, where we synthesize the DNA, and then there's another checkpoint here, and this one prevents progression into mitosis unless we know that the DNA has been fully duplicated and we have all of the proteins necessary to undergo mitosis. And then there's a third checkpoint in mitosis to helpfully ensure that mitosis is proceeding correctly. If any of these checkpoints are not crossed, either the cell will remain in stasis or the cell will undergo apoptosis so as to not become abnormal. It's one of the mechanisms that we prevent abnormal cells from moving on and dividing, creating a tumor. When that cell process gets abrogated, that's one of the ways that cancer cells can proceed in the body. There are several other things that have to happen. You have to be able to evade the immune system, and you have to be able to grow blood vessels and some other things. But these checkpoints are major aspects of preventing cancer from developing. And they're one of the reasons why the incidence of cancer is less than 1 per lifetime. So here I have just the arrows here just showing you these checkpoints and so forth. But now is time to put all these pieces together. What makes binary fission different from cell division, eukaryotic cell division? What's happening in the different phases, generally speaking? We have to receive the right signals, grow the right, express the right proteins, get the signal to pass that checkpoint, synthesize DNA, synthesize the proteins for mitosis, ensure that we have replicated the DNA correctly, undergo mitosis. What's happening in mitosis? Those events and so forth. But it's a beautiful process. It's not super duper technical, but probably a bit more than you might have learned in previous classes. What we do in biology and in clinical medicine is we bring this information to you like layers of paint. So we give you a first coat, and then we give you a second coat, and that second coat covers a little more than the first time. You're going to see this again and again as you go into advanced cell biology, or if you take a cancer class, or if you take a class on developmental biology and so forth in your advanced academic endeavors. In any case, this wraps up part 2 of chapter 12. I will see you again when we talk about sexual life cycles and meiosis in chapter 13. Thanks again, and I will talk to you soon.